everyone at this recap session for the session Novel Concepts of Atherosclerosis from the Vascular Discovery 2021. And I must say it's a beautiful session. It was a beautiful session. And I'm Esther Lutgens. I was one of the moderators, and my other moderator, my co-moderator, was Katey Rayner. We heard several novel concepts of atherosclerosis, and I think they are really important in understanding the pathogenesis of atherosclerosis and have huge implications for novel ways to diagnose and treat atherosclerosis. So one of our speakers who could not join here is Dr. Benjamin Abbott, and he's from the Dana-Farber Cancer Institute in Boston, and he gave a wonderful talk on the role of clonal hematopoiesis in cardiovascular disease. So essentially what he described is that you find these clones of these chip clones of clonal hematopoiesis of unidentified potential, which are precursors for cancer. However, you also find them in an aging population that are otherwise healthy, and not all chip patients convert to cancer patients. And what he, among others, discovered is that these chip mutations are really great predictors for cardiovascular disease. And you also could see it in the biology, because if you have these mutations, you get more clones, especially of the myeloid population, and you actually had an increased risk of cardiovascular disease. So I think that this is a very novel, important concept. We need to learn a lot more on the pathogenesis, but it's really important that if you age, you get these clones and they don't lead to cancer, but they are a real good predictor of having cardiovascular disease. So screening the age population for these clones and these mutations could be a future diagnostic for predicting cardiovascular disease. So that was pretty much his talk. He went into details, he went into several mutations, their meaning, and he has some beautiful papers, some beautiful reviews out there. So I would say just read them, and I'm sure you're convinced of this novel concept of atherosclerosis, and this is one of the future tools we're going to use. So Katey, do you want to proceed?
Yeah, so another great talk that we had was by Dr. Ebru Urbe, who's from UCLA, Cedars-Sinai in Los Angeles. And she gave this very interesting talk about the way that macrophages sense cellular stress and how that really leads to dysfunction in the macrophage and the immune cells and leads to atherosclerosis. And what was really interesting about her talk was actually the sensor, the cell stress sensor that they found is actually in a gene called FMRP, which has really only been linked to Fragile X syndrome. And this is not historically a syndrome that you think of when you think of cholesterol and, you know, coronary disease, but it goes to show that there are so many genes that we just don't understand what they do, especially in the context of metabolic challenges like high cholesterol or you know, obesity or other things. So her talk was really sort of diving down into these mechanisms of how these genes that we're learning about through all these beautiful genetic studies, how they might actually be functioning to cause disease. And so speaking of genetics, one of our other speakers was Cristen Willer from the University of Michigan. And her talk was an absolute incredible tour de force about using genetics and human genetics in particular in different ethnic groups and how that it can inform us about disease and risk. Can you give us a couple of highlights of your talk, Dr. Willer?
Happy to. So I was providing this update on behalf of the Global Lipids Genetics Consortium, and we have a manuscript that is accepted in principle at Nature, so that will hit pretty soon, coupled with public release of all the summary statistics, ancestry-specific summary statistics related to the 5 lipid traits that we examined, LDL cholesterol, HDL, total cholesterol, triglycerides, and then non-HDL. And so the update was really a deep dive into a human genetic genome-wide association study, meta-analysis of 1.65 million individuals recruited from over 200 primary studies from 44 countries. And that gave us not only power to detect association, which is better power than we really had before, but it also gave us the chance to compare what we are studying more diverse ancestries than we have in the past. And while the total number of loci that we can identify is generally driven by sample size more so than diversity, ancestry diversity really drives the ability to find map at these loci, to hone in on functional variants and functional genes, and also very strongly improves our ability to predict individuals at high risk of LDL cholesterol and presumably at high risk of other atherosclerotic diseases from a wide set of ancestries. So being able to have polygenic scores that are more equitable and more effective in different populations was really a benefit of this multi-ancestry work.
Well, congratulations. It's beautiful work, and I think all of these sessions have really highlighted how our field is moving on beyond just identifying genes, but actually functionally understanding what they mean for disease, because that's really where we need to go, is, you know, beyond just understanding. I mean, the human genome brought us where we are, and then ENCODE has brought us even further, but you know, now we're really into understanding, you know, what does this all mean? And of course, once we know that, we start to be able to either tailor existing therapies or come up with new therapies. And I think that's really what this will be all about. This was a great session. Thank you so much, Dr. Willer, for joining us. And thank you to all of our speakers, Dr. Urbe and Dr. Ebert as well. And I think we had a great discussion in our attendees.